Hello everyone, so this is a recap on photosynthesis, on what we've done so far. So you know that plants, when they are exposed to light, which will bring solar energy if it's in the case of the sun, we have this reaction. So carbon dioxide and water are transformed into glucose and oxygen. This is an equation that you need to know by heart, and we'll see later on that we will bring a little bit more complexity to this equation, but this basic equation, carbon dioxide plus water gives glucose and oxygen, you need to know it. Okay, so what we'd like to focus on now is where does photosynthesis take place, and we're just going to recap the detail we've got. So we started off with these iodine experiments. We know that in the green parts of leaves, when there is light, starch is produced. So we can narrow it down to green parts of leaves. If we do an experiment with a microscope, we can then narrow it down to chloroplasts. I'm just going to go and use my pen software and I'll be back. So as I was saying, green parts of leaves to start off with. When we do the same experiment with iodine and microscope, we then know that it occurs in chloroplasts. Starch is produced here when there is light. You've got a bit more knowledge about chloroplasts now. You know that this organelle has this specific structure. This type of diagram, you must be able to reproduce it, so a two-dimensional version. This type of diagram was obtained by looking at this type of picture. This picture is taken with a TEM microscope, so transmission electronic microscope, because we can see the inside of the specimen. And the electrons are going through the specimen. If it was a SEM picture, a scanning electronic microscopes, where the electrons bounce off, we would only see the outside, which is not the case here. We can see the inside of the organelle. You can probably recognize the granums here, and they are made up of granule thylakoids. So we've got one here, we've got one there, and in between these granule thylakoids we have intergranule thylakoids. So photosynthesis, we can be even more precise, will actually occur in these thylakoids, because in the membrane of the thylakoids we have photosystems. Photosystem 1, photosystem 2. They are structures which have many pigments, and it's these pigments which will use the energy of light to enable photosynthesis to occur. We have something called ATPase. We'll see later on that this produces ATP, produces energy. We'll see how, but later on. Okay, let's just zoom on these photosystems. I'm just going to move to the next slide. So in these photosystems we have different pigments organized in an antenna with the various pigments, chlorophyll B, carotene, xanthophyll, and a reaction center which is made of a chlorophyll A. So this is what I want you to know for the moment, is that photosynthesis occurs in thylakoids of chloroplasts, and we'll see exactly how electrons go through chlorophyll A molecules. So they're going to be a part of more detailed mechanisms that we'll look at later on. But as I said, we're building this gradually, so for the moment you need to know the equation of photosynthesis and that it happens within the thylakoids of chloroplasts. Two other things we can just cover. So I didn't say it, but the aim of this video is just to do a few recaps in maybe a different way than I did in the lesson, just for you to memorize in a different way. So we talked about factors which influence the rate of photosynthesis. The first one is temperature. This is because enzymes are used in photosynthesis, and enzymes have an action in photosynthesis, and therefore, as you know, enzymes operate with a specific temperature, an optimal temperature. So if it's too cold or too hot, the enzymes will not work very well. So you need the optimal temperature. Second thing, light. The energy for photosynthesis comes from light. So if there's no light, there is no photosynthesis. And if you increase the light intensity, you can see that photosynthesis increases. Second thing about light is that depending on the wavelengths you have, that will also have an impact on the rate of photosynthesis, because the pigments will absorb the different wavelengths in different ways depending on the different plants. Last thing, CO2 concentration. CO2 is a reactant, so if you put more CO2 in the medium, that means more reactants, and that means that more products at the end of the day. So more CO2, more photosynthesis, and more products such as glucose and dioxygen. And last thing we talked about, first thing before last, so chromatography techniques. So we take, to start off with, we have a green leaf, we then crush it, grind it with a mortar and pestle, we add organic solvent, then we extract. So this is the filtered extract, which will then be placed at the bottom of our filter paper, and when the solvent goes up, this will separate the different pigments, so chlorophylls, carotenoids and xanthophylls. Very similar to electrophoresis, because the denser, more heavy molecules will stay at the bottom, whereas the lighter, less dense molecules will go towards the top. It is possible for scientists to calculate a retention factor value, and this enables scientists in specific conditions to know exactly what pigment they have. So by doing the calculations, by dividing where the pigment stopped by the solvent front, you'll have the retention value. So chlorophyll B will have its own, chlorophyll A will have its own, etc. That enables scientists to know exactly what pigment they're dealing with. Last thing we talked about is the difference between an absorption spectrum and an action spectrum. So quite similar in some ways. The common points. We have on the x-axis the wavelengths, so ranging from 400 to 700 nanometers, and going through the different colors: violet, blue, green, yellow, orange and red. However, on the y-axis it's quite different. Here we have the absorption of the different pigments. So what is absorption? It's the capacity of a pigment to absorb wavelengths. To calculate the absorption, we use a spectrometer. So what we can see here is that with an empty cuvette, the light goes through and we measure a normal intensity of light. With the cuvette filled up with this purple liquid, we can see that the light intensity has diminished. This is because the pigments in the solution here have absorbed some of the light. That's why the light intensity is lower. So what scientists do is that they're going to send through this cuvette all the different wavelengths, so 400, 401, 402, etc., and measure a certain absorption for a pigment and place the different measurements on the graph. This is interesting because we can see that the different pigments will have different absorption rates. If I sum up, chlorophyll A will basically absorb these wavelengths and these ones, whereas, for example, carotenoids are mainly going to absorb these wavelengths. Okay. Action spectrum now. So action spectrum, we have the rate of photosynthesis. This can be measured in dioxygen, and this can be measured looking at the concentration of dioxygen which is produced. So same thing, you need to expose the plant to different wavelengths and simply measure with a probe the concentration of dioxygen. So for this plant here, we can see that photosynthesis is efficient for these wavelengths and for these ones. So last thing, we can compare these two graphs, and there are some similarities. If we look at the outlines of the graph, we can see that we have a similar pattern for both graphs. If we add up the effect of all the pigments, we roughly have the same outline. What can we deduce from this? We can deduce that the rate of photosynthesis is linked to the presence of these pigments. If these pigments would not be present, photosynthesis would not occur. So it's these pigments which enable photosynthesis to occur. And to have this particular photosynthesis efficiency, you need the cumulative effect of all the pigments. So when you have chlorophyll A, B and carotenoids, photosynthesis will be efficient for all of these wavelengths. This is the recaps I wanted to do on photosynthesis so far. So here we are, and see you for the next video. Goodbye.